We're now starting the third section of our Streptococcus lecture, and we are going to start out by talking about another species of strep, and this one is Streptococcus pneumoniae. In the clinical setting, Streptococcus pneumoniae is commonly referred to as pneumococcus or diplococcus. The characteristics of Streptococcus pneumoniae are that it is a normal inhabitant of the nasopharyngeal areas of healthy individuals. It is an opportunistic organism, so when the opportunity arises, it will cause disease, but in a normal healthy individual, it doesn't commonly cause disease, and it will produce a capsule. So that's the main virulence factor of Streptococcus pneumoniae is capsule production. The capsule is antigenic. There are over 80 serotypes, and again, capsules prevent phagocytosis. Strep pneumoniae can also produce toxins such as pneumolysin, and pneumolysin generates partial hemolysis of red blood cells. The more common diseases caused by Streptococcus pneumoniae, of course, like its name indicates, it does cause pneumonia. It's the most common cause of community-acquired bacterial pneumonia, and it's much more common in individuals with chronic underlying disease. It can cause meningitis. It's one of the 3 leading causes of meningitis. It will lead to upper respiratory tract infections such as sinusitis and otitis media, and it can also cause endocarditis, arthritis, peritonitis, so it can cause many different diseases. Streptococcus pneumoniae has a capsule, so you can see a clear halo around the organisms in this slide, and usually capsules don't allow stain to penetrate into it, and that's what you'll see, a very clear halo around the organism. Here you can see a tissue sample, and you can see the capsule, the clear area around the organism. Now, a key characteristic about Streptococcus pneumoniae, which is why it's commonly referred to as diplococcus, is it forms a diplococcus. 2 cocci, gram-positive, that chain together in pairs. So again, the microscopic morphology these are paired gram-positive cocci, and the organism in its pair tends to be more lancet-shaped. So instead of being perfectly round, it kind of flattens up a bit. On media, such as sheep blood agar, it is a smooth, glistening, wet-looking mucoid organism because it does produce a capsule, so it tends to be very mucousy. It is alpha hemolytic, so you'll have that greenish tinge to the agar medium around the organism. And Streptococcus pneumoniae has enhanced growth in CO2. So any specimen that might contain a streptococcus, such as a sputum specimen, tends to get incubated in the CO2 incubator in the clinical laboratory. Streptococcus pneumoniae, of course, it is a strep, so again, it is catalase negative. It is susceptible to optochin, and optochin, like bacitracin, is a disc. You drop it on you streak a lawn of bacteria, you drop the disc on, you stick the plate into the incubator, and then you look for a zone of inhibition around the disc or no zone of inhibition. So Streptococcus pneumoniae is susceptible to optochin, so you should see a zone around the disc if Streptococcus pneumoniae is in the sample. It is bile soluble positive. This is a Streptococcus pneumoniae in a fluorescent stain from a spinal fluid, and you can really see in this stain that lancet kind of flattened shape of the Streptococcus pneumoniae in its pairs. Here's the identification scheme. This is an alpha hemolytic Streptococcus. You would do an optichin disc, and if the optichin disc has a zone of inhibition around it, it would be susceptible and it would be indicative of Streptococcus pneumoniae. If the organism grows right up against the optichin disc and it's an alpha hemolytic organism that is catalase negative, which would be indicative of a strep, you'd need to do other tests. And we're going to talk about these other organisms and the other tests coming up in the next couple of slides. We're moving on to group D, Streptococcus, and in this group there are the Enterococcus organisms, the Enterococcus genus, and the non-Enterococcus Streptococcus species. So the group D Streptococcus and Enterococcus are facultative organisms. Of course, they're gram-positive cocci. They tend to chain up, but again, in this gram stain, you can see they're somewhat clustered, so you can't always tell the difference between staph and strep under a microscope. Here you could see chained organisms, and Streptococcus tends to chain up more when you do a gram stain of a direct clinical specimen, or you grow the organism in a broth. If you take a loop and pull a colony off a plate and then gram stain it and it's a strep, you don't see those nice chains like you would in a broth specimen. Streptococcus and Enterococcus are catalase negative and oxidase negative. The group D streps are normal flora of the respiratory tract of animals and humans, and some of these organisms are found in the gastrointestinal and urogenital tracts in humans. They can cause opportunistic urinary tract infections. They can lead to bacteremia, endocarditis. They can also cause wound and soft tissue infections. And Streptococcus bovis, which is a group D strep, has been associated as causing gastrointestinal tract cancer. So if Streptococcus bovis is recovered from a blood culture, that individual should very quickly be scanned for GI neoplasms to see if they have a GI cancer. The more new concern about the Enterococcus is vancomycin resistant Enterococcus, and that type of organism is incredibly difficult to treat. The organism tends to elongate under the microscope, so some of the group D streps won't really look as circular or cocci. They might be flattened out ever so slightly. Most of the group D streps and the Enterococcus species are non-hemolytic. However, there are a few that can show either alpha or, in rare cases, beta hemolysis. And again, the group D organisms possess the group D Lancefield antigens on their outer surface. So for lab diagnosis of the group Ds, the catalase of all the streps are negative. The group D streps can sometimes produce a very weak catalase reaction. So I put a link up in your course website so you can look at a catalase reaction. And a positive catalase, there's no doubt about it. It is bubbling like crazy. But with a weak catalase, like a group D strep, you might see a little bubble pop up, bloop, bloop. Unless it's bubbling like crazy, if you're trying to differentiate between a staph and a strep, it's likely catalase negative. If it's bubbling like the video, then it's catalase positive. The group Ds will hydrolyze bile esculin, and the way to differentiate the group D Streptococcus from the Enterococcus species is using a 6.5 NaCl, agar slant, or a PYR test. So for your identification flow chart, if you have a catalase negative, gram positive cocci, it would be indicative of a strep. If it is non-hemolytic, you might be thinking a group D strep. So what you want to do is do a bile esculin test. If it is positive, you then need to do your 6.5 NaCl test to differentiate your Enterococcus from your group D. So Enterococcus can grow in the presence of 6.5% NaCl, and group D strep cannot. If the bile esculin test is negative, it's just another type of non-hemolytic streptococcus, and it's not a group D streptococcus or enterococcus species. The Viridans streptococcus is the next group, and you remember these organisms do not have Lancefield antigens, so they're not in that group A, group B category. They are in their own group called the Viridans group. These inhabit the oral and nasopharyngeal tracts of humans. Again, they lack Lancefield antigens. Most of the Viridans streptococcus species are alpha hemolytic or partial hemolytic, but there are some that can be non-hemolytic, and the Viridans streps are the most common cause of subacute bacterial endocarditis. So the Viridans streptococci are located in the oral cavity of humans, so they're normal oral flora. What happens is individuals that have, heart disease or have chronic conditions, if they have any type of dental work, that organism can commonly get into the bloodstream and then get into the heart and cause severe heart damage and bacterial endocarditis, which can lead to, serious chronic disease of the heart and possibly death. Streptococcus, the Viridans streptococcus can also lead to abscesses, mouth abscesses, brain abscesses due to the location of the organism in the human body. So Streptococcus versus your Enterococcus organisms. So you have your hemolysis on blood agar. You always want to look, is it alpha hemolytic, beta hemolytic, or gamma hemolytic? If it's alpha hemolytic, you are differentiating between Streptococcus pneumoniae and Streptococcus viridans. And the way to do that is through biosolubility and the optochin test. If it's beta hemolytic, you're considering Streptococcus pyogenes, or group A strep, from the non-group A streps, such as Streptococcus agalactiae, and that you want to do your bacitracin tests for. If it's non-hemolytic, it could be either Enterococcus or group D Streptococcus, and you want to do your bile esculin and 6.5% NaCl. The general characteristics of all of these Streptococcus species: the pneumococci, or Streptococcus pneumoniae, and the Viridans streptococcus, they are both alpha hemolytic organisms, and both are part of the normal upper respiratory flora. So you're going to find these in the oral cavity, the oropharyngeal cavity of the human respiratory tract. Group B and D Streptococcus are vaginal and intestinal normal flora. Transmission is usually direct contact with the mucosa, droplet secretions, which can lead to pharyngitis or sore throat, strep throat, direct skin contact, poor hygiene, and sharing inanimate objects in an area of trauma. So you have a serious abrasion and there's something nearby that's contaminated. Control and prevention, hand washing is of course the number one way to prevent transmission of infectious organisms. Also avoiding contamination of your Streptococcus agalactiae during delivery. So Streptococcus agalactiae is normal flora of the genital urinary tract of women, of not all women, but a lot of women carry it. So part of the pregnancy evaluations and prenatal screening and all those things is to determine if an individual is a carrier of Streptococcus agalactiae. If they are, they will be treated for it to reduce the risk of having complications due to group Streptococcus agalactiae during delivery. Treatment, a lot of these strains can be treated with penicillin. To treat Enterococcus, it's resistant to penicillin, so ampicillin or other types of non-penicillin based antibiotics or antibiotic combinations. Vancomycin resistant Enterococcus has a very high mortality rate because it's so difficult to treat. For pneumococcus, the cephalosporins beyond the second generation and the quinolones are more commonly used. And now we're going to move on to the final lecture of the Streptococcus.